Our brain works like a computer and neurons do the computation. So it is called neuronal computation. Now in this video, we are going to learn about one neuronal computation, that is neurons make an all or none decision to generate an action potential. In laboratory, we can insert a microelectrode inside a neuron and then we can connect this microelectrode with a battery. And if we do this, we can stimulate the neuron by injecting current. We can also insert another microelectrode inside the neuron to measure the resulting membrane potential change that occurs due to the injection of current. If we do this, we will get a graph like this. Now, before we understand this graph, we have to know three important points. The first is resting potential. In resting neurons, that means in an unexcited neuron, the membrane potential is called resting potential. I have made a dedicated video on resting potential. You can also watch. Now, if the membrane potential becomes less than resting potential, we call it hyperpolarization. And if the membrane potential becomes greater than the resting potential, so we call it depolarization. Now, in the first injected current, the membrane potential of the neurons actually becomes less than resting potential. So it causes hyperpolarization. And we observe that no action potential is occurring. In the second injected current, the membrane potential goes below than the previous, so it also causes hyperpolarization, and we observe that no action potential is actually generating. So it is for sure that hyperpolarization do not actually generate action potential. Now, if we change the polarity of the current, a neuron actually responds accordingly. Here, the membrane potential actually increases above the resting potential, so it causes depolarization. But we observe that no action potential is also generating here. So this three situation is called passive electrical response when action potential do not occur. Now, in the fourth injected current, a full firing of an action potential is observed. Now, what determines that whether an action potential will occur or not? And that is by this red line called threshold potential. Now, if the membrane potential during this stimulation meet or exceed the threshold potential, here it is minus 50 millivolt, then a full firing actually occurs. And if not, no firing at all, and that is called all or none. So action potential is actually an active electric response. Now let me briefly explain what happens during the passive electric response. As you can see here, the membrane potential of the neuron actually decreases as the distance increases from the site of current injections. And if you minutely observe, you will find that this decrease of the membrane potential occurs within a range of less than 1 millimeter. Now, what is the reason behind this decrease of this membrane potential? The reason is because the currents actually leaks out from the action. And the conclusion is that passive conductance actually decays over distance. Now, the prevention of this passive electric response in neurons is very important, because in circuits there are neurons called interneurons, specifically interneurons that are present in the brain circuits. They have tiny actions that length about 1 millimeter or less than that. Now, if this passive electric response is not prevented in this interneuron, then this interneuron can send the electrical signals to the other neurons, and that can cause a noise in the brain. In case of active electric response, what we observe that it is completely opposite to the passive electric response. Here the membrane potential remains constant, so we will get a straight line. So the conclusion is active conduction is constant over distance. Now, active response via action potential actually circumvent or overcome the inherent leakiness of the neurons. Now, as we have understand that there is a threshold potential, so there must be a threshold sensor. And in the active response, we have found that once an action potential is generated, the membrane potential remain constant all along the length of the action. So there must be a booster system. So we will actually learn these 2 points more in details. Voltage gated sodium channels. This channel actually remain closed at minus 70 millivolt. That means at resting potential of a neuron. Basically, these voltage gated sodium channels contain voltage sensor domains. There are 4 voltage sensor domains present in a voltage gated sodium channel. I'm only showing 2. This voltage sensor domain, or in short VSD, contains many positively charged amino acids. During resting condition of the neuron, the inside of the neuron remains negatively charged. So these positively charged VSD domains actually remains very close to the cytosolic site of the neuron, and that causes the close-in conformation of the channel. Now, what happens when depolarization occurs? Within a range of -50 to 0 millivolt, all the voltage gated sodium channels actually open, because the VSD or voltage sensor domains actually sense an electrostatic pulling force towards the exoplasmic surface of the neuron, and that causes a change in the conformation of the gate. And as because there is already a sodium concentration gradient is generated during the resting potential generation of the neuron, the outer concentration of sodium remains high than inside. Through channels, ions move always from higher to lower concentration. So sodium actually comes inside of the neuron. Now, what about the booster system? Basically, this booster system is actually a positive feedback loop. The greater the initial membrane depolarization occur, more voltage gated sodium channels actually open, because voltage gated sodium channels acts as threshold sensor. They can sense the change of voltage. So if more voltage gated sodium channels open, what happens? More sodium ions actually enter inside the neuron, and that causes more depolarization. One important point is that when sodium enters inside the neuron, the inside become more positively charged. Excess positively charged that present in the cytosolic side of the neuron and excess negative charge that are present in the outside, they move in both directions. This is not by diffusion but by settling of charge, and this is called passive flow of current. The ions that move through the open voltage gated sodium channels is called active current. So through the voltage gated sodium channel, passive current and active current actually flows. Now that causes, this passive flow of current causes opening of additional voltage gated sodium channels in the adjacent segment of the neuron, and that causes the positive feedback loop, because as more sodium channels open, more sodium enters, more depolarization occurs, and then another segment of the sodium channels present gets open. Now the question I want to ask here, that the passive flow of current actually moves in both directions, but we know that action potential move in one direction, from action to the action towards the action termini. So why not the action potential move in both directions? I will give this answer in my next lecture. Now up to this, what we have learned is in laboratory situation. In physiologic conditions, the neurons receive information from other neurons. Up to tens of thousands of neurons form synapses with one neuron. Now synapse mean the physical junction between two types of neuron. The neurons that send the signals called presynaptic neuron, and the neurons that receive the signal called postsynaptic neuron. Here the yellow colored neuron is the postsynaptic neuron. Basically the cell body and the dendrites are the main structure that receive the information, and this white color button like structure are the axon termini that are sending by this presynaptic nerve. Now synapses could be either excitatory, that means it can help to trigger an action potential, or inhibitory, that can discourage the action potential. Here the green dots represent excitatory synapse and the red dots the inhibitory synapse. Basically two proteins like PSD95 and geoffrin are actually tagged with two different fluorescent dye. These two proteins, PSD95 and geoffrin, are the scaffolding protein. They helps in the clustering of neurotransmitter receptor. The PSD95 helps in the clustering of glutamate neurotransmitter receptor, and glutamate actually acts as a excitatory neurotransmitter. And geoffrin actually helps in the clustering of gamma aminobutyric acid or GABA neurotransmitter, or glycine, that acts as a inhibitory neurotransmitter. Basically the excitatory synapse causes small depolarizations in the postsynaptic membrane, and the resulting membrane potential change is called excitatory postsynaptic potential, or in short EPSP. The depolarization occurs due to the opening of voltage gated sodium channels through which sodium enters inside the neuron, and also opening of voltage gated calcium channels through which calcium enters. And as because these ions are positively charged, so it actually reverses the membrane potential, and at the resting potential the inside becomes negatively charged, so it becomes positively charged, so it causes depolarization. In case of inhibitory synapse, it causes small hyperpolarizations in the postsynaptic membrane, and the resulting membrane potential change is called inhibitory postsynaptic potential, or in short IPSP, and it is occurred due to the opening of voltage gated potassium channels through which potassium ions come outside of the neuron, and as because potassium is positively charged, and when it moves out of the neuron the inside become more negative, so it causes hyperpolarizations. Also opening of voltage gated chloride channels through this chloride ions can enter inside the neuron, so it also causes the insight more negative, so it causes hyperpolarization. Now, individual PSP, that means individual postsynaptic potentials, specifically the excitatory postsynaptic potential, do not fire action potential, because we know that a neuron are receiving synapses from up to 10,000 of neurons. So a single synapse could not actually generate action potential. There are mainly two reasons. The first is individual PSP raise the membrane potential only a few millivolts that do not bring the postsynaptic membrane to the threshold potential. As we have learned that threshold potential must have to meet or exceed. Another reason is that the plasma membrane of dendrites and the cell body contains few voltage gated sodium channels. As you can see, the dots here present is the sodium channel. In the cell body and the dendrites, there is low amount of voltage gated sodium channels present, but a segment called axon hillock where the voltage gated sodium channels is highly dense in condition. So the axon hillock is very sensitive to the change of membrane potential. Now, what happens in physiologic conditions? Summation of excitatory and inhibitory postsynaptic potentials occur. So neurons spatially average excitatory and inhibitory PSP and generate an action potential only when their sum at a particular time brings the membrane potential to threshold in the axon hillock. In the axon hillock, there is high concentration of voltage gated sodium channels present. Now let me explain how the summation occurred by taking this simple example where only 3 synapses has considered. See here, the resting potential is minus 60 millivolt. Now if we activate the excitatory synapse like E1 or E2, the membrane potential actually increases above the resting potential. Now when we activate both the E1 and E2, both the excitatory synapse, the membrane potential actually meet or exceed the threshold potential and it causes a full firing of action potential. Now the inhibitory synapse, if it is activated, it causes the lowering of the membrane potential than the resting potential, so it causes hyperpolarizations. And in case of when we activate the E1 with the inhibitory, we find that the membrane potential actually now decreases. Also, we can observe that when all these 3 synapses are activated, E1, I and E2, no action potential is occurring due to the inhibition of this inhibitory synapse. So neurons make an all or none decision to generate an action potential by mainly summation of excitatory and inhibitory postsynaptic potentials. And there is a threshold potential. If the membrane potential change meet or exceed the threshold potential, the voltage gated sodium channel that acts as a threshold sensor, it gets open, and that causes a positive feedback loop or booster system.